Hello everyone, my name is Romina Melo. I am a PhD student fellow al Grupo Ecología de Poblaciones de Insectos in Bariloche, Argentina. The main title of my study is Worker Reduction Affects Investment in Reproduction in Eusocial Wasps, a Case of Tax Flexibility. In social insects, division of labor is common. A few individuals reproduce, queens and drones, whereas the rest of the colony, the worker caste, performs different tasks within the colony. Workers can specialize in different tasks such as brood care, nest maintenance, and foraging. Worker task specialization in social insects is a factor that has determined their success because it allows optimizing invested workforce. Also, flexibility in allocation of individuals to different tasks allows the colony to respond to external changes in the environment and internal perturbation, such as that arising from differential mortality of individuals undertaking a given task. We evaluated experimentally in colonies of Vespula germanica whether the removal of workers affected reproduction, measured as the number and quality of genes. In addition, we analyzed some colony tasks such as protein food collection, defense, and hygiene that could affect investment in genes. We went to the field, searched for wasp natural nests, we removed them off the ground, and placed them in experimental boxes. After an acclimatization period, different percentages of workers from 0 to 76% were removed from this nest. Then, periodically, we collect the amount of protein food entering the nest, count the number of workers showing a cleaning or defense behavior, and capture genes leaving the nest. For each gene was calculated the body mass index, which is the ratio between the weight and the natural logarithm of the length of the tibia. This index was used as an indicator of quality for the genes evaluated. Through a general linear mixer model, we show that the number of genes produced was affected negatively by worker removal. Similarly, removal of workers affected negatively the amount of protein food collected. We didn't observe a significant effect of the quality of genes, the number of workers devoted to cleaning or guarding the nest. These results suggest that wasp colonies could be producing fewer genes in response to the decrease in protein food collecting. Wasp foragers collect protein from animal prey to feed their developing larvae, and the larvae need to reach a minimum weight for pupation. Therefore, the availability of food at this stage is a key factor. However, gene quality was not affected by worker removal, maybe due to the fact that weight and size are a critical factor for survival during wintering of genes. On the other hand, number of workers showing a cleaning or defense behavior was not affected by removal, maybe related to the small sample size analyzed or an existence of a flexibility in allocation of individuals to different tasks. Understanding task allocation within the worker case could provide a better knowledge of the mechanisms that promote the success of invasive social insects. Thank you very much for listening to me. If you have questions about my work, you can contact me at the email that shows up on the screen. You can also find more information about Grupo de Ecología de Poblaciones de Insectos by scanning the QR code.